This is part 3 of our lecture on Chapter 17, and we've talked so far just about transcription, which is taking the DNA information and putting it into the language of a messenger RNA. And now we're going to focus on translation of that information into an actual protein. So translation is occurring from the RNA nucleic acid language, which gets translated to an actual protein language, the amino acid language that builds our polypeptides. And that starts with looking at our messenger RNA in terms of its 3-letter codes, or the triplet code that we call a codon. So during translation, the messenger RNA is actually read in triplets called codons, and it's read in the 5' to 3' direction. So you can see there, for example, where UGG is a codon for tryptophan, that amino acid. And again, this should be reviewed from your general biology class. This is just another picture showing that same idea of taking the DNA, transcribing it to messenger RNA, and then translating that into a sequence of amino acids. All of this comes down to what we call the genetic code, which is not really overstated in being called the code for all of life. It's the strongest support for a common origin for all of life because it actually works in all sorts of organisms. The code is redundant, which means that several codons will code for the same amino acid. There are 64 codons. They were deciphered in the mid-1960s. And of those 64 codons, there are 61 codes for amino acids. There are 3 codons that act as stop signals to end translation as well. And then we all have our 1 start codon, which is the AUG, which codes for methionine. The last point here in this list is that the 3rd portion of the code is called the wobble position. And we'll talk about what that means. But actually what it does mean is that if you look at the different things that will code for the same amino acid, oftentimes what's different in the codes is just that 3rd of that triplet there. So the 3rd nucleotide that's there is the one that is the wobble. So what that actually, if you look at leucine's a really good example where we have all 4 different possibilities, U, C, A, and G, in the combination of the first 2 CUs. So that 3rd wobble position there really shows us how there can be room for mutations or errors in the code when it occurs in that 3rd position, because the 3rd portion of the code seems to have some flexibility to it. And it's shown in the way that we have this redundance among the different codons. So codons have to be read in the correct reading frame or groupings in order for the right polypeptide to be produced. And again, this should be review, but that codon has to be read in the right order and with the right reading frame, which means reading the correct 3 together as that codon in order to make the correct amino acid. When we talk about the genetic code being nearly universal, what we mean is that it's shared by the simplest bacteria to the most complex animal, being us, and that genes can be described or transcribed and translated after being transplanted from one species to another because of this universality of the code. So the same three bases together are going to code for the same amino acid whether they're in a bacteria or they're in a pig. Whether they're in here, you can see what's occurred in these pictures is the transformation of what you're going to be doing, actually putting a glowing set of instructions from a jellyfish into different organisms. And you're going to be doing that this week by putting it into a bacteria to make it actually glow. But you can also take that same sequence of nucleic acids or nucleotides and put them into a tobacco plant and have them, in this case they're using firefly, but you can have it cause this tobacco plant to glow. But that same one, the pea glow that we're using with bacteria, they've actually put into the pig, which has caused his snout to glow and his hooves to glow. So that is just again showing the universality of this genetic code, where you can code for the right protein, in this case a glowing protein, take it out of a jellyfish and put it into a pig and still get that same protein created. So we have the same translation ability between organisms. So this picture shows the basic concept of translation, starting with the functioning of the ribosome, the presence of our messenger RNA, and the use of the transfer RNAs to bring in our amino acids to build our polypeptide chain. There are two steps needed for accurate translation. The first step is a correct match between the transfer RNA and an amino acid, and this is accomplished with an enzyme there called amino acyl tRNA synthetase. So this amino acid is responsible for matching up the transfer RNA with the right amino acid. Next, you need a correct match between the transfer RNA and its anticodon region with the codon that is found on the messenger RNA. So if you get these two steps done correctly, then you get the correct placement of an amino acid onto the polypeptide that is being synthesized by the ribosome. And again, that wobble idea is defined as a flexible pairing at the third base of the codon, and it's called a wobble because it allows some transfer RNAs to actually bind to more than one codon. The structure of a transfer RNA molecule has been described as a clover leaf. So if you look at it two-dimensionally, it does have this clover leaf idea. And you can see it is a sequence of nucleotides. In this case, they're RNA nucleotides. And they're in a polynucleotide sequence. What we think of when we think of a transfer RNA is the portion at the end here that attaches to the amino acid to bring it over to the ribosome, and this portion, which we call the anticodon, which matches up with the messenger RNA's codon. These are some other depictions showing the idea of the transfer RNase molecule. And this one gives you the more three-dimensional idea. But you still can kind of see a little bit of that clover leaf But important for you to remember is that every molecule of tRNA is specific for the type of amino acid that it's going to bring. So the anticodons are going to be different between the different tRNAs, and the sequence here that attaches to the amino acid is going to be slightly different as well. So transfer RNAs are not identical molecules. This is that single-stranded RNA view of a transfer RNA. And you can see that a tRNA molecule is about 80 nucleotides long. When we flatten it, you can actually see the base pairing that occurs. A transfer RNA molecule does look like the clover leaf, and you can see that it's held into this basic shape with some hydrogen bonds in between the different nucleotides, similar to what we see in our double-stranded DNA. But remember, like messenger RNA is not double-stranded, and RNA is not a double-stranded molecule. But in the case of transfer RNA, it does have some portions that look double-stranded, and those double strands are base paired in the complementary bases bonding together there. So you see the cytosine guanine there, the uracil and the adenine. Because of these hydrogen bonds, transfer RNA actually twists and folds into that three-dimensional molecule that we saw in the previous slide. So again, to load the transfer RNA with the correct amino acid, there's the need for this enzyme, aminoacyl tRNA synthetase. And that enzyme will bond the amino acid to the transfer RNA. And this requires energy. So ATP is actually required to load the transfer RNA with the amino acid. So you can see here, this picture is being shown of the use of the enzyme, this aminoacyl enzyme here, which is helping to take the substrates of the transfer RNA and the amino acid and bond them together. And you can see that's a dehydration reaction. We have water being produced because there is that actual bond being formed. So now moving on to the ribosome. So the destination for that tRNA is going to be the ribosome, which is made up again of rRNA and proteins. And the ribosome's job is to facilitate the binding of the tRNA anticodon to the messenger RNA codon. And actually, think of it not so much as binding as really matching up. The structure of the ribosome is two subunits, one being large and one being small. And there are three sites located on a ribosome. There's the A, P, and E site. So the A site is the one that holds the transfer RNA, which carries the next amino acid to be added to the chain. And then the P site holds the transfer RNA's growing polypeptide chain. And then the E site is the one that empties the transfer RNA and allows it to be released to leave the ribosome. So it's called the exit site. There are two populations of ribosomes present in cells. And we talked about this back when we were talking about cell structure a little bit. But just to clarify and make sure that you understand the difference, we have free ribosomes that are in the cytosol, and we have bound ribosomes which attach to the endoplasmic reticulum. And the free ribosomes are really the ones that do most of the synthesis of proteins. and they function in the cytosol. And the bound ribosomes make proteins of the endomembrane system and proteins that are secreted from the cell. But these bound and free ribosomes are identical, and they switch from being free to bound. So much of the attachment of a messenger RNA to a ribosome, most of it occurs in the free ribosomes floating in the cytosol. And once that occurs, if there is a need for that new polypeptide chain to be processed within the ER, then that free ribosome will go over and attach itself and become a bound ribosome in that rough ER. So you can say that polypeptide synthesis always begins in the cytosol, and then it finishes in the cytosol unless that polypeptide signals the ribosome to attach to the ER. And these polypeptides that are destined for the ER or for secretion are actually marked by a signal peptide located in, or created from that messenger RNA's amino acid sequence. This is a picture again showing the different players here in translation, and we have the two subunits of the ribosome shown here, and how we will get the messenger RNA, which will bind to these subunits and cause them to actually form one unit. And then what will happen will be the attraction of these transfer RNAs, bringing their amino acid into the first site, moving over to the second, and then being released in the third, and how that occurs through the matching of the anticodon to the codon. And when we have the amino acid attached to the transfer RNA, we call that a charged tRNA. So it's an amino acyl charged tRNA when it has its correct amino acid attached. Just like in the building of our messenger RNA, we have the three parts of the translation process. We have the initiation, elongation, and termination of a growing polypeptide. So initiation is when we have the messenger RNA, the ribosome subunits, and the initiator tRNA with the start codon all come together and start or initiate the translation of the protein or polypeptide. Elongation is the process or the time when we're adding amino acids based on the codon sequence, and then the termination is brought about by one of those end codons. So this is just going to show you initiation here. During elongation, amino acids are being added one by one to the C terminus of the growing chain. And each addition involves proteins called elongation factors. So we have codon recognition, peptide bond formation, and then translocation. These are the three steps in elongation of the polypeptide. So again, the first step is codon recognition by the tRNA, a peptide bond formation joining the new amino acid to the peptide chain, and then translocation of the tRNA to the next site. So from the A to the P site, and then from P to E through exit. the termination. Going back, one other point I wanted to bring out that I missed on an earlier slide was that there is an energy expenditure at 2 points of this translation process. So we did talk about the first one in terms of the addition of the amino acid to or the loading of the amino acid onto the tRNA. But there's also energy required in translocation and codon recognition. So you can see here there's a GTP required, and we break one of the phosphate bonds in order to get energy there. And we'll see a little bit later on here. The second requirement of a GTP to GDP is when we're translocating the tRNA along from site to site. So there are 2 GTPs needed for each addition of an amino acid to our polypeptide. So a termination occurs when the stop codon in mRNA reaches the A site of the ribosome. The A site will accept a protein called a release factor, and that release factor causes the addition of a water molecule instead of an amino acid. This occurrence and reaction is going to then release the polypeptide, and the translation assembly comes apart.